So this is a big day, and I'm really pleased to be able to introduce Jeff in his final seminar for his PhD thesis. He's really been an important contributing member of the lab, and his intellectual depth and scholarly approach, I think, really stands out for graduate students. And I think we've all learned a lot from him. He's done a beautiful piece of work that is very complicated and creative and moves into new territory that I think is quite important. So this is a really great time to celebrate what he's done. I want to point out a couple of other things that I think you all need to know about. You mentioned the glowing eyes. So it's kind of a giveaway because we know Jeff, of course, better than most of the rest of you here. And there is another side to Jeff. Next slide. So we're really careful when he starts to move into this. But it doesn't happen very often. Unfortunately, there aren't very many signs telling us it's going to happen. So we tread kind of carefully every now and then. And I'm hoping that this will stay in the background during the talk. So with that, no more points to make. I'll sit down very carefully and sort of hide here on the side. Thank you.
So thank you, Patsy. Thank you, everyone, for coming to my exit seminar to hear about my research. My name is Jeff Eunice. I'm a PhD candidate in the Berkeley UCSF bioengineering program and a member of Patsy's lab. My talk consists of two parts. The first part is on prediction of protein function using a probabilistic model that performs analysis of sequence similarity networks. And the second part extends that model to additionally try to automate analysis of genomic context. So first, some background. By the way, this is a talk with an audience of people from a wide variety of backgrounds. So it's going to be a very challenging talk because as part of my exit seminar, I want to try to go into as much depth as I can because the contributions I made are kind of in the details. So please excuse this very non-technical introduction or non-precise introduction. The genome is the blueprint of life. It's written in an alphabet of 4 biochemical letters and consists of genes and other structural elements. Sometimes the genes encode protein sequences, like this one, and are folded up into proteins. Proteins have different roles. Many of them are molecular machines called enzymes. Here we have the protein sequence for mandylate racemase, and 8 of those get folded up and put into this molecular machine, and its function is to flip the stereochemistry of this hydroxyl. So it just flips on this molecule, it just switches this OH from being in the background to the OH being in the foreground. Determining the function of proteins is necessary for understanding life, and it's valuable in applications in health and industry, including proteins from humans and from other species. There are 90 million protein sequences in a sequence database called UniProt, and this plot shows its growth over time. However, according to a database of protein functions, only 563,000 of them have a function annotation that resulted from experiment or from manual curation. So that's way down here, and the growth of that would look pretty much flat if we had plotted it. The goal of function prediction is to help bridge this gap by electronic annotation. To give a flavor for a canonical method for prediction of protein function, one thing you can do is take the sequence of a query protein. You can search a database of protein sequences that have some functional annotation, and then you take the most similar protein or the most similar proteins, and you transfer over that functional annotation. We're going to try to improve on that method. There are many different representations for protein function. The one that I find the most useful is the gene ontology, which provides a controlled vocabulary of functional terms arranged hierarchically, with more general terms at the top as ancestors of more specific terms. The the gene ontology, models three aspects of protein function. The molecular function, which is defined as the activities that occur at the molecular level. The biological process, which is defined as the series of events accomplished by one or more organized assemblies of molecular functions. And the cellular component, which is defined as the location of where the protein carries out a molecular function. Now I'm showing just three functional terms here, one for each of those aspects and their ancestors. But in actuality, there are 50,000 functional terms defined in this ontology. And here's a fun fact to introduce. The depth of a Go term, which is defined as the maximum path length from the root to the Go term, so this depth would be 2. It ranges from 0 to 17. There is only one Go term at depth 17, and that's calcium export from the mitochondrion involved in positive regulation of presynaptic cytosolic calcium concentration. And there's no proteins that are annotated to have this function experimentally, which is actually quite a common issue. So in the Babbitt lab, we study the function space of mechanistically diverse superfamilies. One of the tools that we use is sequence similarity networks. Here we have a result of analysis of a sequence similarity network of the nitroreductases by Eyal Akiva, right over there. In this figure, each node, each circle, is a protein or a cluster of similar proteins, and each edge represents pairwise sequence similarity. Only the large nodes actually have either a functional annotation, an experimental annotation, or a solved structure. So many of these, the colors were as a result of Eyal's analysis, but that's not the input. That's the output after doing this analysis. So Eyal used the network, experimental annotations, sequence alignment, structures, trees, and other functional data to help draw these functional boundaries, to be able to figure out where there might have been a change of function, and also to identify unexplored regions of the function space. Like over here, there is no functional annotation or structure, but based on other functional data, Eyal determined that it probably does not have the same function as these. So one concern is the data here are noisy, and of course, it has uneven coverage. There are some clusters that are extremely well studied and some that aren't studied at all. So in order to try to solve these problems, I'm going to take a network like this is kind of a high level, imbue it with a probability model in order to try to find the most likely explanations of what each of these proteins are doing, while handling the peculiarities of the problem. So a probability model, like any model, it's a simplified description of how something works. So if it's a good model, hopefully it can be used to make predictions. A probability model is one that can also model error and handle noise in the data. This is particularly good for predicting protein function because it's good for dealing with sparse, noisy data, because modeling change of function in a superfamily is noisy. And we are interested in quantifying the uncertainty in the results, so giving confidence for our predictions. So in order to come up with a probability model, what's required is that we have a way of representing the joint distribution. That is the probability of a given functional assignment to all the proteins in the network. And if we can do that, then we can calculate anything that we're interested in, such as the probability that a specific protein has a specific activity, given that protein has other activities and other proteins have other activities. So it's trying to find a consistent explanation for what's going on probabilistically. However, note that parameters are really important. Here we have a model for the number of heads observed when you flip a coin 10 times. And we present that probability in analytical form right here with a parameter pi. If pi is 0.5, then the probabilities you get are what you'd expect from flipping a fair coin. However, if pi was 0.99, then what this model would actually say is that the most likely sequence of flipping a coin 10 times would be seeing all heads every time. So it's really important how you set your parameters. The framework that I'm going to use is called probabilistic graphical models, or PGM. It has all the advantages of being a probability model, but it's also a naturally good fit for structured and relational data like our protein networks. Probabilistic graphical models allow you to build up a model from smaller components. So here I'm showing that the joint distribution is represented by a product of a bunch of factors over subsets of the variables in the network. And the particular form of those subsets of variables is that depends on the type of model, the semantics of the model, and the assumptions that you want to make about the data. So if you have a directed probabilistic graphical model, then each of its parameters is simply the probability of a particular variable given the parents of that variable in the network, in the graphical model. So all we really have to do to solve this whole function prediction problem is just define these smaller components of the model and also figure out how to calculate the parameters that are reasonable and give us good results. So let's say, as a toy example, we're starting with just two proteins, a network of two proteins, protein A and protein B. And those two proteins are related by sequence similarity. So they have similar sequences. So the first thing we're going to do is we're going to model the molecular function of a protein with a hierarchy of Bernoulli random variables taking the value of 0 or 1. It's going to look a lot like the gene ontology, but instead of modeling all 50,000 terms, 50,000 Go terms, we're only going to model all the terms that exist in the protein network. Here I'm only showing 3 candidate terms. So we saw only these 3 terms in the network. But usually there are a lot more. This is just an example. So one of our query proteins resulted in a template with 504 candidate functions for just the simple model for each protein. So each protein has 504 Bernoulli random variables built inside of it. There are several reasons for wanting to use a hierarchy of Bernoulli random variables in our representation. Compared to a multinomial, we can predict that a protein has multiple functions. A multinomial technically assumes those functions are mutually exclusive. It is possible for annotations to more general functions to be useful for predicting lower level functions. So it can actually benefit if we model like this. We can actually define the relationship between this Go term and this Go term, and they can actually inform each other, which is really important because of how sparse the data is. Also, it's really good for predicting valid structured output labels, so labels that are that make sense with the structure of Go, as well as being able to consume input labels that also have missing data. And finally, by modeling each one discretely, we can add relationships between other variables in the graphical model. So the second thing we do when two proteins are similar in sequence, what we do is we draw an edge between the corresponding molecular function terms. What this does is add a factor that says that two proteins that are similar in sequence are likely to have similar molecular functions. So now that we've defined what the model looks like, the structure of the model, that structure defines what form our parameters have. So as I mentioned before, a directed probabilistic graphical model has its parameters, the probability of a variable given its parents. So the first thing we're going to try to do is get it into a directed form. So to do that, we take the protein network, we make a minimum spanning tree of that protein network. We root it at the query and direct it outward, and then we prune it. So then we end up with a reduced network that's directed and nice and concise. Now the parameters, as I mentioned, are here. And for this model in particular, we're looking at the probability of a particular random variable. So the probability of the parameters are the probability of a Go term given values to its parent Go terms, as well as to its network parent corresponding terms. So all we have to do to get our joint distribution is to multiply all these. This is going to be one function. we're just going to multiply them all together. For every variable, it's going to be a factor that includes this and values to these two. And we just multiply it together, get our answer, and get great results. So one thing that's really one of the most important things about this model is that we can actually compute this from all the data, all the experimental data in the database that we have. So here's an example of such a calculation specifically for the Go term thiol ester hydrolase activity. Here we have, so we're just building a table of the different combinations. So these are counts, and this is counts when it is a thiol ester activity. We have the parent activity, and we also have the parent Go activity, and we also have the neighbor annotated to the same thing. That would be here. That happened 19 times. So basically, we looked at 43,000 neighboring protein combinations, and these are the counts that we end up with. So these are just all combinations of these three variables, and then we count them. So if we wanted to just compute the background probability of being a protein being a thiol ester hydrolase, then all we do is we take all the times that the protein was positively annotated to thiol ester hydrolase, and we divide by the total number of observations. And we get way less than 1%. However, these are the parameters that are incorporated into the model. This is the probability of thiol ester given values to the parents. So what this says is if we know that the protein is a hydrolase activity acting on ester bonds, and we know the neighboring protein is has thiol ester hydrolase activity, then the probability of it being a thiol ester hydrolase activity can now be calculated as 19 plus 19 plus 3. So that goes up to 86%. So besides having these interesting parameters, what they tell us is this is going to add a lot of information to the model about how neighboring proteins and their functions relate. Besides being able to have specific parameters for each Go term, which is a novel contribution, it also allows this directed probabilistic graphical model, semantics, also allows us to do things like add data-derived pseudocounts. So a lot of terms, as I mentioned, don't have any experimental data. So most people think, well, we're not even going to predict on it. In fact, when the community does evaluation, they don't even predict on them because all statistical methods would fail in this situation. However, what we do is we add pseudocounts that are based on similar terms. So for example, we have our modified contingency table, which is equal to the raw contingency table that we just talked about, plus the pseudocount table. And the pseudocount table is derived from pseudocount is derived from a contingency table of similar terms that have more data and more complete annotation. So it's derived from the contingency table of the parent term for that Go term. And you can see how these are the counts after we have the pseudocount, we add the pseudocounts. So you can see by adding more complete information, this is not an accuracy or anything, but by adding more complete information, we now have the probability of a thiol ester, given its neighbors are thiol ester, is now a 98%, which kind of looks more complete, but obviously this isn't evaluated yet, so it could be much worse. One thing we noticed about our model, which results directly from the way that a directed graphical model factorizes, is as shown. Say we have just one protein in our network with evidence, and this is the whole model. It's just one protein, and this is the template within it. So if it's annotated to if the protein's annotated to a cyclase activity, then we know it has the more specific function of having a catalytic activity. However, because of our model semantics, it says that the probability of oxidoreductase activity is independent of cyclase activity, given we know it's a catalytic activity. So what we get directly from the parameters is, in this situation, our probability of oxidoreductase activity goes up to 12%, which is not good because that 12% is going to be higher than a lot of very specific cyclases. And we don't think it's likely that it does both oxidoreductase and cyclase, but because we factorized our model to make it computationally tractable, we end up in this situation. So we handle this two ways. One way is the blue way. And what it does is it takes the top-down model and it adds supplementary negative evidence. So in this case, as I just said, we can look at the data and we can say, well, the probability of it being an oxidoreductase, given it's a cyclase, is really, it's less than 1%. And since that's unlikely, why don't we just force this down? Why don't we say it's experimental evidence for not having oxidoreductase activity? So what we're doing is, with the top-down model, we complete this annotation in a way. And that's one workaround for handling this issue. Another way to handle the issue is to do something that people would think is crazy and make flip the arrowheads around. Now, the result of this is the semantics are the same. So we still have to represent this graphical model for the probability of a term given its parents. But now the child term in the model is catalytic activity, and it depends, and that factor now depends on all of those child terms. So our factor is now a lot larger, and it's now less tractable to solve this problem. But the good news is we don't have to include these assumptions about the negative evidence. What happens on the calculation on the right is observing it's a catalytic activity only would make this probability go up. But that's explained away when we also observe that it's cyclase activity. So what happens if you work out the math is that the probability of being an oxidoreductase, given both these, actually just goes back down to the prior probability of being an oxidoreductase. And that's a really cool benefit. It's called explaining away in these models. But you rarely see a tree-like structure represented in a bottom-up fashion because it's much less tractable. And we're going to see the implications of that later. But we have two approaches for solving this. So now that I talked all kinds about parameters and little modifications, this is something that a few slides that tie the method all together. So the first step is we take a query that's represented by this diamond gray node. Gray means it doesn't have an annotation, and that's it. The diamond means it's a query. And then the first thing we do is we blast for similar sequences. That means we search a database using the tool blast for similar sequences. And we set a pretty high threshold for this, and we bring in all the sequences that are fairly similar. So the purpose of this method, which I haven't mentioned, is we're really trying to differentiate like specific catalytic activities rather than make general suggestions of very general terms. So we collect all the similar proteins, and then we build a sequence similarity network. We build a sequence similarity network by using another tool called Diamond, which is a lot like BLAST, that can do all-by-all comparisons very quickly. So we take this unstructured network and we turn it into a protein similarity network, a sequence similarity network. Step 2 is we construct the probabilistic graphical model. So to get the topology of the graphical model, we take the protein network from the previous slide, we take its minimum spanning tree, we direct it outward from the root, and we prune it. And then we get this topology. You can see in the dark edges, the dark directed edges are actually the minimum spanning tree edges. And the light edges, which are light and it's hard to see, are all the edges that don't really contribute to the model. I just use them for laying out the network so you can see how they compare. Remember that within each one of these little proteins, each node over here is a protein, but within each of these little proteins, it's actually a template that we have that whole hierarchy of Bernoulli random variables inside, and we relate those to the neighboring proteins and within themselves. So this looks like it is because this is a very small portion of that candidate ontology. The final step, which I like to call step 3, is infer protein functions and visualize. So this is 2 visualizations of that previous protein network. The one on the left is colored by the probability of the blue function. And the function and the network on the right is colored by the probability of the red function. And you can see that the network is able to combine evidence from around the network to be able to guess that this query is actually supposed to be blue and not red. And not only can you color the network by any particular Go term, but you can also get detailed predictions for every Go term in the candidate Go ontology. I'm sorry I don't show the whole Go ontology here, but it would be difficult to see. Here you can see the blue one is shaded, and the red function would be somewhere else in this DAG. So evaluation of methods for protein function prediction is very complex. And that stems from having incomplete gold standards, structured label space, imbalanced classes, experimental bias, and even time dependence of the annotations themselves. So to help standardize evaluation, a community experiment is held every few years called the Critical Assessment of Function Annotation, which works as follows. The organizers release a large set of sequences for which they want you to predict, they want the different participants to predict annotations. And then the teams all individually try to make predictions using whatever data that's available that they want. They can do their own experimental data, they can just use BLAST, they can mine the literature, they can incorporate all the different information into a SVM or do whatever they want. And they submit their predictions. And only once they submit their predictions do the organizers start collecting new experimental annotations into the database, new proteins with experimental annotations. And then after they've collected a bunch of them, they evaluate the predictions to see which of them came to pass. So now that we have a method, now that we have a general framework for evaluating protein function prediction methods, question is how can we pair a prediction P with a database multi-label T? So P is the prediction, T is the answer. So fortunately, this is easy. We just look at P and T and we see if they're equal. But that is absolutely not actually okay. The reason is because, as you remember, P is a combination of 50,000 Go terms. T is also a combination of 50,000 Go terms, and we know it's incomplete. Even though it's our answer, we know it's incomplete. So the number of times P would actually equal T is not very likely. And what you get is just every method would get a 0. And that's just not informative to how well the methods are doing. So what people can do instead is look within these multi-labels, and they can look at each term in Go and count how many times the particular term's value matched the value that was in the annotation database. And then divide by the total number of terms. But that is not okay either. And that's because in general, a protein has very, 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 very few functional annotations. So if you were to just make a method that predicted it doesn't have that function, you would get 99.99% accuracy. So that's not so helpful. So what people use in this situation is metrics called precision and recall, which basically just only look at the class of interest when you have an imbalanced class problem. So it just so precision is defined as the number of terms you predicted correctly over the total number of terms you predicted. And the recall is defined as the total number of terms predicted correctly over the total number of true terms that were in the answer. And you can see that there's probably a trade-off depending on how many terms you predict. Unfortunately, that too is not okay. And that's because these don't take into account the hierarchical nature and the uneven distribution of the information of all the terms. So for example, if there was a method that shied away from predicting specific terms and only tried to predict general terms that were very common, would have an advantage. And since the specific terms are the terms of interest, we don't want to bias it in favor of not predicting those terms. So the metric that we use, and is starting to become widely used in the field, is based on precision and recall, but it's an information content-based version. So rather than just summing up, rather than just counting the number of times you got the prediction right, it sums up overall the terms that you got right, the information content of those terms. So if the only thing you do is predict terms that have no information content, like the root term that says has a function, that would just keep adding essentially a bunch of zeros to your prediction accuracy. And then similarly, it's normalized by the same things as before. So it's normalized by the total information content of the precision is normalized by the total information content of what you predicted. And recall is normalized by the total information content of what's in the answer. There's one more caveat. We've talked about how to compare for a single protein a prediction and a true annotation. But there's an issue where not the quality of the annotations in the database are not equal. So someone could come in, take a new organism, and then very shallowly annotate it as everything that's having binding. Then if anyone went in and predicted anything more specific than binding, all those methods would do terrible again. So the last thing is to weight the samples themselves when computing the performance of a method overall, weight the samples that go into that average by their information content as a proxy for the quality of that annotation. So now we can show some performance and see how things look. Almost. Let me just explain the axes here. So a method is going to give a probability for each Go term for each protein. A user normally could then take only the highest confidence predictions, or if they're looking for, casting a wide net, then they can take more of the predictions in order for increased coverage. So here we have the two axes of the last metric I showed, the sample weighted precision and the sample weighted recall. I'll just call them precision and recall. And the plots are generated by calculating precision and recall for each probability threshold for the predictions. And what you get is a method can, if you set a threshold of 100% for a method, it'll get great precision, but its recall will be pretty much 0. Conversely, you can set a threshold of 0% and say, just give us every Go term in the answer, every Go term that exists, and it will go down to pretty much no precision, but you'll get all the recall. So the goal is to be as far to the upright as you can, which is good precision and good recall. But again, we want to focus more on the high precision parts because we're really trying to differentiate catalytic activities, not look for broad answers, not for general functions. So here I've implemented one of the baselines. This is a form of the naive method. So the naive method typically just doesn't even look at the protein and just predicts the background distribution for all the Go terms. So if the term binding happens 50%, it'll get a probability 50% for every protein. Naive plus is a modification where the first thing it'll do, it uses that Bayes net that we have the parameters for, and the first thing it'll do is it'll take all the annotations that are in the training data as certain, and then it will, I'll just give an example. If a protein is annotated in the training data as having polymerase activity, and we know that 50% of polymerases are DNA polymerases, then we'll have a prediction of 50% for DNA polymerase, and we'll get 50% probability for RNA polymerase as well. So it uses the sample data and it uses the probability of child term given the parent terms that we know, and it makes the predictions that way, but it really doesn't use anything about the protein sequences themselves. Notice that if the protein that you're looking at doesn't have any annotation in the training data, then it would be exactly the same as the standard naive method that just uses the background probability distributions. One thing to point out is that This analysis here is an aggregate analysis over all proteins in the evaluation data set, even if the methods were unable to come up with any annotation because they failed to do inference for any reason or failed to make the network or anything like that. So since naive method, well, you'll see. So here we have two more baselines. This is a blast-based method. And the idea of a blast-based, it's using the same, sorry, the mouse is on that is different than the one I'm looking at. Which is very confusing. The blast-based method, again, searches for the database. It collects all those homologs, which is the same as the first step in our method. And then it transfers them all directly without building a network, just by transferring them based on the normalized score of how similar those proteins were. So if it hits itself, because it was in the training set, it'll transfer that annotation to 100%. And all those annotations for that protein to 100%. If it's 90% similar to a protein with some annotations, all those annotations will get transferred at 90%. Now, this is the random blast method shown for scale. Now, it's actually what this method does is it takes the same set of homologs, the same set of related sequences, and when transferring the probability of those annotations, rather than looking at the normalized blast score, the normalized similarity score, what it does is it just chooses a probability uniformly at random. But this is actually not a bad method. The reason it's not a bad method is because our blast search in the first step only consists of very closely related homologs. So all the functions that are being transferred over are very reasonable. Second of all, it's almost doing some type of enrichment analysis, because when if it's hitting multiple proteins and those proteins all have the same annotation, experimental annotation for the same Go term, then because it's choosing a probability, because it's choosing its probability uniformly at random, what's going to happen is there are going to be more chances for those terms that occur frequently to have a high probability. So the biggest limitation of the random blast method is probably that it can't tell when it's hitting against itself. Even when it hits against itself, it's still transferring that with a random probability. You can imagine that maybe if it was using its own training data, maybe it would start over here, and then the difference between between blast and randomly ordering the blast results would actually be even slimmer. Here are the two methods. Here's the one method. Carroll 2006 presents the method that's most similar to this work that's published in the literature. It doesn't do all those it doesn't do things like the negative evidence and the supplementary. It doesn't use things like the supplementary negative data. It doesn't make things more tractable by doing the minimum spanning tree, and it doesn't use similar parameters. And you can see that it has low coverage and low accuracy. Carroll 2006 Plus is the same as our method of fusion, except it uses their parameters. So it still uses all the things that we could use, all the little benefits that we added, except it uses their set of parameters, which I don't think are likely to be the maximum likelihood parameters. They're just they chose a non-principled set of parameters, which was necessary based on the semantics of the model they used, which I can't get into too much. But the point is, we perform better than the most similar methods published in the literature. Now, one of the top performing methods that does automated phylogenetic analysis is called Sifter. And this is not my implementation in any way. I used a published pipeline and a published version of the algorithm. It does things very differently. It makes homologs based on all the PFAM domains that are in the protein. But you can see this is one of the top performing methods, and it only performs actually a little bit better, at least in this scenario, for this evaluation, only a little bit better than random blast in this very critical evaluation. But this is stuff we're still looking into. Oh, I forgot, I haven't even mentioned performance R methods yet. These are the performance R methods. So it outperforms blast, the previous published methods, the previous published most similar methods, as well as the top performing methods in CAFA. And you can see that we perform better than blast, better than all these methods. Now, if we focus instead on just the treated proteins, that is the proteins that our method was limited in only having 40 minutes to make its answer for each protein. And in the previous plot, if it failed, then we just gave it, it didn't help its prediction. So BLAST would do great and our method would do terrible. So this only looks at treated proteins, which we call, we define as proteins for which all the methods that are plotted here were able to make a prediction. And you can see our method, our top-down method, performs the best. We also did a per protein analysis. This is an actual example. So we did a per protein analysis in order to identify good and bad cases between a fusion and BLAST. And in this particular case, BLAST, which does not use the network and cannot see the clusters, actually predicted the query had the red function, because apparently this red protein is more similar than any of these blue proteins. So our method was able to pick out that it should actually belong with the blue function. And of course, the simplification, we're actually talking about a whole hierarchy of functions. Where's the mouse? Sorry, I'm looking for it. I'm sorry. Yep. So there are lots of other results. I just presented the main results here, but there are a bunch of other results. We also looked at, for example, the performance on catalytic activities, but those were similar, so I'm not going to show them separately. We can calculate, we can quick and reliably generate sequence similarity networks. And we did all kinds of really fun analysis. How well does adding a network help and that kind of stuff. One thing to note is that PGMs are a natural fit for this problem, but we're really pushing their limits. As you could probably see from the bottom-up model, where it didn't perform as well, that was likely due to the additional computational complexity. So there's some really cool directions for future research for this first part of the talk. They're straightforward, but they're really interesting. We only looked at proteins, when we collected homologs, we only collected homologs for proteins that were that could be aligned across the entire sequence. But we're also really interested in looking at it from a domain perspective. So the whole point is that our model can have fine-grained measures of similarity, and we can actually know when two proteins share a domain and only propagate the function that is represented by that domain. And that's something that we think would be really cool. Also, of course, we can model additional aspects of functions such as cellular component. Cellular component, there's a lot of high throughput data, and knowing if a protein does its stuff inside the cell or outside the cell has a big effect on the types of molecular functions that are likely to occur. Finally, we can use this a probabilistic model for a lot more than just computing marginal probabilities. One obvious thing would be to use it for target selection. We can calculate the variance of the model and see which proteins we should subject to further study in order to gain the most knowledge or reduce the uncertainty the most in our superfamily. So that's the end of part 1 of the talk. This is the beginning of part 2 of the talk. Now we have a predictive framework that does some rudimentary sequence similarity network analysis while taking into account the peculiarities of protein function, like its hierarchy, the incompleteness, that kind of thing. And we know that there's very little experimental data, such that we need to incorporate other types of data to be able to help draw these functional boundaries. I talked already about how we can use structures, multiple sequence alignments, and other techniques, but the one that I'm going to try to integrate now is automation of genomic context analysis. So the example that inspired this work is shown here. It's from Frank Rochel's lab at Texas A&M. Here we have a query protein, and we have 3 proteins that are similar in sequence. Because these are all annotated to be nucleotide deaminases, the query was also predicted to be a nucleotide deaminase. But when they looked at the pathway, the other proteins that occur nearby in its genome, they noticed that they are all involved in histidine degradation. And it wouldn't make sense that there's a nucleotide deaminase in the histidine degradation pathway because histidine is not a nucleotide. So they actually thought about it for a while and hypothesized and validated that actually it is essentially a deaminase of a histidine substructure. And it turned out to be the right answer. Even if, so that's the size of, this is good research, but consider the case where we have something more complicated. You might think that if none of the homologs are annotated and none of the proteins in its operon or genomic context are annotated, you might not be able to get any information about the query. But if you expand the context and look at the network, you might be able to think, you might try to work out how we can find some explanation given these proteins of what's happened, of some information about the query, some information about what might be going on. The only problem is this is starting to get difficult, and the actual scale of the problem looks something more like this. And that would be very difficult to do manual genomic context analysis of. I've been talking so far about genomic context. That's one type of functional association that says that two proteins do some work together. But there's other types of functional associations too, such as such as two proteins being in the same metabolic pathway or have some type of be involved in a multi-protein complex. And there's a lot of these can be detected through computational experimental assay. And we use 46 million of the highest confidence functional annotations, which combine this data into a database called string. So when I mention string, I'm talking about predicted or experimentally verified high confidence functional associations that we got. So now I'm going to quickly show how we extend the previous model of fusion into a fusion GCA, which can additionally incorporate this functional association data. This should look pretty similar because it's the same as a fusion. And the reason why I left that space there before is because now when we have two proteins that are functionally associated, we're going to associate their biological process terms. So now we're going to model a different aspect of protein function, which represents its process or pathway. And then say that those propagate, we're essentially saying two proteins that are functionally associated are likely to have the same biological process function. Now, what we end up with here is essentially a bunch of disjoint networks. The molecular functions can't talk to the biological processes. So we complete this model by associating highly correlated terms between molecular function and biological process. The first thing you might have noticed, actually, I might be able to skip over this part. One thing I'll show is this is an actual candidate. This is an actual ontology of candidate Go terms. The white Go terms are from the original model. And now that we include functionally associated proteins, we have these additional functionally associated molecular functions. And we also model an entirely new ontology, entirely new aspect of function. So the blue ones and the yellow ones are all in this model. And as a result, it's much more computationally intensive. And plots show that we're starting at a disadvantage compared to the previous method because we have a much more complex model. Skip over those. So just like before, the parameters of the model, it's now the third time I'm saying this part. So I'm sure you all know everything about Bayesian networks now. But the parameters are still the probability of a specific random variable given the parents of that random variable in the network, in the model. So originally we had this parameter, this set of parameters. And now we have two other sets of parameters. One is the probability of a term, of a biological process term, given its parent biological process term and its corresponding biological process term in a functionally associated protein, in the most functionally associated protein, actually. And we have this interontological parameter, which is the probability of a Go term given its parent Go term and its interontological parent term. Looking at the specific parameters, you can actually see that they are very useful. So here we have, this is saying if we know a protein, basically the background probability of it being a carbohydrate biosynthetic process is 7%, but we know it's functionally associated to a protein that's involved in carbohydrate biosynthesis, then that probability would go up to 78%. We also have to figure out which cross-ontology associations to mine. I'm going to pretty much skip this, but the idea is you collect a bunch of examples where we collect a bunch of examples in this novel way, and then we find the metabolic process that shares the highest mutual information, basically the highest correlation to the catalytic activity that we're looking at. And we do this given its parent term. And that's so that we can really find terms that we can really find associations that will help say that break these ties for us and really figure out the specific protein functions. And you can see some of the ones that were found. Catalytic activity was most associated with metabolic process. That's a good starting point. Nucleotidal transferase activity was most associated for the particular model we were looking at with heterocycle metabolic process. And these are the correlations. And I can't read this one, so I'll let you read it yourself. And again, if we look at the actual parameters that are calculated based on these, and again, we're mining these from all the available data in the data set, which is, again, something that you couldn't do with the previous models, with the semantics of the previous models. But when we learn it from all the data, we can see that essentially what this is saying is that given we know a protein is a nucleotidal transferase, it's 30% likely to be an adenyl transferase. But if we also know the protein is involved in organonitrogen compound metabolism, then the probability that's a nucleotidal transferase increases to 85%. So there's a lot of information both in the functional associations we're adding and in the interontological associations we're adding. And again, if you forgot what we were doing here, we start with these input annotations, we run inference, and we get some output annotations, as well as detailed predictions for every protein network. So unfortunately, it did not improve the results over the previous method. I think a lot of that from our detailed analysis is that the model is much more complex. And even though the model is much more complex, I didn't give it any additional running time. I held the running time for inference to be fixed at just 40 minutes. So as you can see, I'm guessing that at this point, we're still just copying over the training data, and we see a gap as it wasn't even able to predict what was in the training data. And then it did fine, but we think it started at a big disadvantage because of the additional computational complexity. We did a per protein analysis comparing a fusion GCA, which includes the functional associations, to the original version of a fusion. And in this case, a fusion predicted the wrong function before the right function, but after including the functional associations in the biological process, a fusion GCA was able to predict the right function before the wrong function. And of course, I can go into details about which functions those are, but in the interest of time. Again, looking at the per protein analysis, we looked at the proteins that contributed the most to the error in those plots. And the top 6 of them, just by searching, all were uncertain that they even had the root molecular function term, which means inference failed for these proteins, and it couldn't complete it in 40 minutes. Even the 7th protein, all the probabilities looked really weird. They're all either 0 or 1. So therefore, we think a reason for a fusion, again, in addition to the previous plot, looking at detailed analysis makes us think that a reason for the decreased performance compared to a fusion was that it just didn't have enough time for some of the more complex, for some of the query proteins with more complex networks and candidate ontologies. Also, I keep showing these networks as actual full networks, but one fun thing to see if you only use the model is only based on the minimum spanning tree. So you can also lay it out just based on those edges, and you actually get something that looks like this. So you can't see the clusters anymore, but the minimum spanning tree, the edges in the minimum spanning tree actually usually stay within the clusters. So it works well. Other results from the second part are that we can create and use these sequence similarity functional association networks very quickly. That's helpful to a lot of people because it shows the useful annotations among functionally associated proteins that might help people who do manual analysis. We can use the model that we have now to flag unlikely combinations of annotations. So we can use it as a quality check in a database of annotations, where if we see a molecular function that doesn't make sense with the biological process, we can flag it. And one thing we didn't even talk about or evaluate is that, in addition, a lot of labs are interested in predicting biological processes. And while we were interested in using biological process to predict molecular function, we can also get information. Our biological process predictions should also be pretty good. And in the CAFA, the preliminary results, this was one of the top performing methods for predicting biological process, and we weren't even focused on that. So there are directions for future research that are really cool. One thing that is kind of obvious is to continue this idea of automating genomic context analysis. One thing we could do is incorporate substrate substructure information and similarity and docking experiments as evidence. We can also automate other aspects of genomic context analysis, like when two homologous proteins with similar genomic context are more likely to be orthologs and have the same function, opposed to a protein that has a completely different context. But none of that is currently handled in the model. So conclusions for the whole thing, there were a bunch of contributions. There were the models, methods, implementations, predictions, the detailed analyses about how all the different components worked. We did a rigorous evaluation and provided evaluations. Also, there were other projects that I worked on while I was here. Limitations. I can probably skip limitations, right? No, our model gets much more complex as we're including a lot of different functions. And a lot of electronic annotations might actually be useful because they might be based on a different aspect of analysis. But if we include all the electronic annotations, our template might have all 50,000 Go terms in it. And our model can handle that, can handle that kind of scaling right now. Second, we use a minimum spanning tree, which was incredibly useful, but it can't perfectly capture network boundaries. So it would be useful to look at ways to include more edges, as long as we can keep the representation that lets us incorporate all the parameters and use all the experimental data and to do so. Again, there's a lot of really exciting general directions for future research for this model. We can model other aspects of protein function, similarity, and we can use the model for doing other things besides predicting molecular functions. So this is the last slide, the acknowledgement slide. I really want to thank everyone in my long time I was here, mostly my advisor, Patsy Babbitt, who was a phenomenal scientific advisor and a mentor and a role model. It was wonderful to be in her lab, and I think she made me and everyone else a better scientist in person. So thank you, Patsy. Everyone else in Patsy Babbitt's lab is in blue. There are a few additional members that I had not yet put on there. These are sorry. But I want to thank, I've talked with everyone. They've heard this talk so many times, they could have given it, maybe not the specific talk, but they've gone over every trial and tribulation. So I really want to thank them for listening to so many conversations about parameters. I want to thank the other faculty that have been a big part of my training, particularly Hao Li, who's on my committee, and Andre Shali, who is on my qualifying exam committee. John Holstenbeck at Berkeley is on my committee. I learned a lot from Stephen, who I first worked on Sifter with over at Berkeley. Ian Holmes, I did a lab rotation with him. Kim and Scholander, I TA'd with her and learned a lot of my first bioinformatics from her. And I also reached out to people at other campuses, Ariel Jaimovich and Yishan Chen, who are experts in probabilistic graphical models. I also want to thank my friends who listened to the talk and read over my dissertation, some of them more reluctantly than others. And most of all, my dissertation is dedicated to my family, especially my mom and my dad and my brothers. They were very supportive of me, and stressed out I got, the more they supported me. And the more I neglected them, the more they supported me. So thank you. And of course, my cat was always there to support me as well. So thank you very much, and thank you for your attention. I hope you enjoyed it, and please keep in touch. And thank you.
Question?
So it sounded like you touched on this in future directions, but including ligand information in your model, like what ligands bind to the protein. You expand on that a little bit, how that might work and how that might help your model.
Yeah, the general idea would be to separate out maybe a high-level protein function with a specific substrate and propagate which substrates, so model substrate in each template, and then consider propagate which substrates are likely by looking at substrate binding site similarity. And then also you could relate which functions, which substrates are likely within which biological processes by looking at, you look at all the proteins in a biological process, you can narrow down its substrate substructures, and then you can say whether or not this substrate is likely in this biological process. So it's just relating all these different components and being able to say which combinations of them are unlikely combinations. But that's the idea. So model substructure, the relationships within the template and between different proteins.
So I have a question. Predicting functions is really important, but there are a huge proportion of predicted functions in the current databases that are probably wrong. And when you are able to add genome context, which our lab has done some experimenting with, it's such noisy results that it's very hard to figure out how to filter out the important signals. And you made a comment about being able to flag the sort of not very nonsensical results. And I think that while that sounds like kind of a sideline, it could be a really important way to get a better, both to use genome context more effectively, but also to help people identify where their proteins may have been misannotated. Because if you think a protein does function A and you spend a lot of time doing an experiment making that assumption, and that's not quite right, you're kind of dead in the water, and you've spent an awful lot of time and money in going in that direction.
Yeah.
So I suggest you really consider developing that part of it as well.
Yeah, there's some in the literature on interontological correlations. That's actually one of the main focuses. They don't actually go as far as predicting protein function. They actually are looking at functional associations, and sometimes they use them for curator tools. When curators are annotating a protein function, sometimes it will suggest a term that's always co-occurs with a experimental term that they've seen, and they annotate it or something like that. So it is actually I think this is definitely a novel approach for that, but it is definitely has some momentum behind it.
It's very undertreated though.
Mm-hmm.
Any other questions? Let's thank Jeff again. And thank you all.